We're looking at the LEAF topic paper. Now it's interesting, if you look at this question, right, it's important to know this actually. Look, ignore these symbols. Right, let's that there is layer C, that is layer B, that is layer B, this bit here is layer B, that is layer A, and that is layer C, and then you have at the bottom the lower epidermis. Right, but I just want to show you this, the way the questions are, well, they seem to be changing this way anyway. So look, they've broken it down. They're saying, how does layer A help? How does layer B help? So you've got 5 marks. And they've added, how does layer C help? And how does layer or structure D help? So they've got 10 marks. It's a 10-mark question. Now, the way I've seen things changing is they've turned those into 5 or 6 markers, and they've saying describe or discuss, and then it's for you to break these down into parts. Right, okay, so this here, this layer is called the upper epidermis. Epidermis meaning outside layer, so it's the upper epidermis. Then you've got the here, this bottom layer is called the lower epidermis, and it contains, these are called guard cells, and if you look, there's a little gap here, the stomata. Right, then you've got this layer here, and that's the mesophyll palisade layer. And then you've got, right, look at this structure here. That's a vein, and it consists of a xylem. So I remember it as WX. So water is for xylem, and it contains also the phloem. Phloem, this is how I remember it, F for food, although it's spelled with a PH. And then you've got this bit here, this layer here, the whole layer. So you've got the vein inside that layer, but this layer is called the spongy mesophyll layer. So that's the structure. We need to discuss that. Okay, so just remember that. Right, so the upper epidermis, it's got this waxy cuticle. Right, it stops water evaporating out, and you've got this transparent layer of cells. They are transparent. Because they're transparent, they allow lots of light through. Right. Right, this here, this is a palisade cell, and can you see all these little dots here? They are in effect chloroplasts, right? So you've got, let me bring that here. So that is a palisade cells with lots of chloroplasts. Lots of chloroplasts. Why? It's at the top of the leaf, so it catches the maximum amount of sunlight. Okay, so I'm doing it like this so then you can make a sentence out of that. Then you've got, what have we got? Let's use that. You've then got the xylem and phloem, right? The phloem transports the products, so it transports the products of the photosynthesis process. Photosynthesis. It transports those. Where does it transport those? Around the plant, wherever it's needed. And xylem is for water, because water is needed for photosynthesis anyway. You've got the spongy mesophyll layer. Now, if you notice here, can you see these gaps? Right, there's moisture here as well, because what that does is that allows water to diffuse. So the air gaps, or air spaces, or air gaps, allow diffusion of gases. So it allows gas exchange or diffusion of gases in and out of the leaf. Yeah, so what will happen is when CO2 comes in, it'll diffuse in and it'll work its way, right? And when it produces oxygen, which is a waste gas, it'll diffuse out and then out of the stomata. Right, okay. Right, and then finally you've got the lower epidermis, where you have the guard cells. They open up during photosynthesis to create a pore or a stomata, and again that allows CO2 to come in and O2 and water vapor to come out. Right, and we need to write those as two markers. So this is what's going on. Right, how do we go about doing that? Layer A. The layer A is it's always good to just name them. Epidermis. Don't just put epidermis, because we're talking about the upper, not the lower. Has lots of transparent cells to allow light in. Layer B is called the palisade layer, right, which is densely packed and has lots of chloroplasts. lots of chloroplasts, and is located at the top and located near the top, near top of leaf to capture or to absorb or to capture maximum amount of light. Right, layer C. This is called the spongy mesophyll layer or the spongy layer. Spongy mesophyll layer has air gaps. These are standard answers that you can learn, and just then if they ask you, you apply them. Which allows for gases to diffuse in and out of the leaf. So oxygen will come out, CO2 will get diffused in. You've also got in this layer you also have the it's called vascular bundle. I hope I didn't call it vein. Vascular bundle. I probably did call it vein, but vascular bundle's better. Containing the phloem. Okay, the phloem to move the products of photosynthesis around the plant to other parts of plant. And the xylem, which transports water for photosynthesis, which is an ingredient for which transports water and minerals, for photosynthesis. So remember we said water and carbon dioxide are the two ingredients that we need to produce glucose, and you end up with oxygen as a waste product. Right, structure D was the guard cells. So the guard cells open and close. Done it again. Guard cells open and close when photosynthesis. Okay, let's just stick to open when photosynthesis occurs to allow carbon dioxide in, and you also get it allows water to transpire water out. You also get the waste product of oxygen, and you'll get oxygen out as well. Right, okay, that's cool. Right, water lilies float on the surface of ponds. So like basically what's going to happen with this one, you're going to get you're going to get a water lily, and that's water around it, so it's on a pond. Now that's water, that's your water lily. Now if you had the guard cells here, the stomata would open and you wouldn't get much air in, carbon dioxide or oxygen out. It'd be difficult. So what you notice with water lilies, the stomata are at the top. So gases, CO2 and oxygen and water vapor can get out. So they're saying the structure D, which is the guard cells, are found on the upper surface. What's the benefit on that? So the lower surface is not exposed to air, so no carbon dioxide would be able to get in. Also, no transpiration would take place. And what is transpiration? So that's when the water comes in through the roots, up the stem and out of the leaf. So you've got your roots here, you've got your soil there, you've got your stem there, and you've got a leaf here. So what happens is water comes in through the roots, up the stem and out of the leaf. This here is transpiration. This is the transpiration stream, and it just keeps doing that, and it helps keep the plant cool, especially during photosynthesis. Right, we've got a microscope of the upper and lower surface of a leaf. This shows the lower surface. So can you see that guard cell there? And there's a stomata here. There's a stoma. Right, that's another stoma and another one, and we need to count them. So look, we've got 1, 2, 3, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. Right, suggest how the upper surface of a land plant would differ from this diagram. Okay. On a land plant, you would have so the upper surface of a land plant would differ. You would have fewer okay. So this is the lower surface of a land plant. The upper would contain fewer stomata. Plants make food by process of photosynthesis. Write chemical equation. Right, this has come up before, so we write carbon dioxide plus oxygen gives, what did we say glucose was? Carbon, hydrogen, oxygen, 6, 12, 6. So remember, hydrogen is double oxygen, and you get a waste product of oxygen. And what did we say? This is the big molecule. All the small molecules, you add the 6, 6, 6, and it's done. Right. Right, now what they've got is the leaf of a plant is adapted to carry out photosynthesis. Explain how the following parts of a leaf are adapted for photosynthesis. So palisade mesophyll layer, right? It's densely packed. Densely packed. That means it has lots of chloroplasts. Right? Why? To capture maximum amount of light, and it's at the top as well. It is located near the top of the leaf. Right, spongy mesophyll. Spongy mesophyll layer would be, this has air gaps or spaces to allow gases to diffuse in and right. What diffuses in? Normally CO2 and out. What diffuses out? Normally the waste product, oxygen. Right. And the guard cells, they will open up to form a pore or a stoma, or lots of form stomata, to allow CO2 in, oxygen and water out. Right. This is another experiment that you need to know. The student used this method to show that starch is produced during photosynthesis. It's good to know this, that photosynthesis produces glucose, which is turned into starch. So you put a potted plant in the dark 24 hours. What that does is that removes any starch. It destarches the leaf. Let me go to the side. There's your leaf. Right. It's got starch inside. Starch is, you can look at it as a lot of glucose molecules stored for future use, for energy. Right, when there's no photosynthesis, the plant needs to stay alive. And if there's no glucose being produced through photosynthesis, what happens is this starch breaks down into glucose. And then the plant uses this glucose molecule to respire aerobically to stay alive. And then after about 24 hours, what happens is all the starch disappears. You end up with a destarched leaf. Then put the plant in bright sunlight for 12 hours. Remove the leaf from the plant and put in a beak of boiling water for 30 seconds. That kills it off. Then put the leaf in boiling tube containing hot ethanol for 10 minutes. That removes the color, the pigment, the chlorophyll. Right, you have to be careful. That is flammable. That means it can catch fire quickly. So you're not going to put it over a Bunsen firm. You're going to use it. You're going to heat it in something called a water bath. That, like an electric kettle, you can call it that. Wash the leaf in cold water. That's to cool it down. Put in a white tile so you can see the color change more easily. You add orange, brown, yellow iodine solution. Again, depends on the concentration. And what happens is it will turn blue black or black blue. Right, whichever way you write it, but write black blue. Right, what that does, that indicates that's a test for the starch molecule. Right, so that's what's happened. Now we need to write that down. Explain why the student put the plant in the dark for 24 hours. Right, is to de-starch the leaf. And it also didn't allow photosynthesis to take place. There you go. Right. Describe how student would carry out step 4 safely. So that was boil it in ethanol. Remember we said ethanol is flammable. So do not use a naked flame or Bunsen burner, but use a water bath to heat. Okay. Right. State the color of the leaf. So we said blue black. It turns blue black. It's best to use both the words blue. It turns blue black. Blue black. The student concludes that the starch is produced by photosynthesis in the leaf. State a suitable control that she should use to make a suitable control. Right. What were the three variables? Dependent, independent is the one you measure, and the control. You have control variables. Control variables. These are the ones that you make sure that the test is fair. You're only changing one thing, one variable. What could you do? Where are we? What you could do is leave a leaf in the dark and test using iodine. Yeah, that's what it is. So what you could do is you could have two leaves in the dark for 24 hours and then you take this out, put it in the light, and then test it with iodine, and then test that for iodine and see what happens. Right. Plants need to absorb mineral ions. Magnesium is used for chlorophyll. Right, and then nitrate. What you can do is they'll allow, just say ammonia or ammonium ion. I suppose you'd have to put ammonium if it's iron. So yeah, I would stick to nitrate. Stick to nitrate for proteins, magnesium for chlorophyll, phosphate for nucleuses or nuclei. I think we've done this. Okay, so a student investigates the effect of carbon dioxide on photosynthesis. Student places a plant in the dark to remove the starch. Explain why a leaf becomes destarched. Because there is no, I've done this before, there's no photosynthesis. Because there's no photosynthesis, what happens is starch breaks down or is used to form glucose. So starch breaks down to form glucose to carry out respiration. Not to carry out respiration, for respiration. Right, to stay alive, basically, yeah. Safe method to destarch leaf. We've done this. Testing, so we need to test. Right. Cut leaf. I'm just going to go through this quickly because I think this is the standard thing. You boil it to kill it. Boil it in water to kill it. Make sure there's no naked flame, because you're gonna then heat it or boil it in ethanol to remove the chlorophyll, the green pigment, chlorophyll. Right, you've got a structure here called a chloroplast that contains a green pigment called chlorophyll. Right, to remove the chlorophyll, but you make sure there's no naked flame because you're boiling ethanol, because ethanol is flammable. Then you wash it in cold water, and then you add iodine, and it will turn blue-black. Right, what you could do is add some details of equipment that you're going to use, instruments. So use goggles in case to protect eyes. Use tongs. Use tongs. What do you use tongs for? To stop scalding your hands or burning your hands when you're boiling the leaf and you're trying to wash it. Okay, that's that. Right, so you've got a leaf here. So what I'm going to do is draw a leaf here. Here. So remember, that's that line there. This has got no CO2 because potassium hydroxide has absorbed it. We've done this in another video. Right, because there's no CO2, there's no photosynthesis taking place, even though that there is light coming in, because you need CO2 and water. So this will have no starch. So that will be when you put iodine on, it will remain green, and this outside will be blue black. Okay, so that was that one there. Right, what they want you to do as well is no starch. Okay, rabbits eat grass, cell wall. We need to draw, label other structures in a leaf. Right, let's do this. Let's go with this. So you've got here, you've got that is a cell wall, and inside you've got a cell membrane. So you have to be patient with this. There you go. That is the cell membrane. Right. You've got a big plastic pack of water. That is called a vacuole. Let's write that carefully. Vacuole. Right. And then you've got chloroplast. Should have done that green, really. Should have done that green, really. Green. Are you misbehaving again? Right, just bear 1 second. Okay, didn't seem to be behaving, but it doesn't matter. So that's your chloroplast. That's that. You've got your cytoplasm. That's the liquid part. And what else have you got? Yeah. You have the nucleus. There you go. Right, that's a similar question. See what we've got. Right, okay. We're gonna have to do this. Right, okay. We need to finish this off actually. It's part of the same. No, it's not. Right. We'll create another video for this, inshallah.